Thank you so much for joining us. One of the companies that I'm the most excited about as an analyst is actually a private company, but maybe not for that much longer, and it's Juventas. We have the CEO standing next to me, sitting next to me, Rahul Aras. What I'm excited about also is Mark Penn. Mark Penn is part of the brilliance of this company, and he's a very experienced cardiologist and certainly a key opinion leader in the space. One of the things that you have to look for as an analyst when you're looking at any company is how can one company shift the entire landscape? When we talk about CAR T therapy, when we talk about gene therapy, when we talk about eliciting and changing signals and the profiles in the body, we have to be thinking about Juventas. One of the things that's going to be required for cell therapy to work most of the time is that the cells have to know where to go. They have to understand what to follow. And I think that when we look at Juventas and we look at the technology and the science of this company, it challenges me to think that, one, this is a very enabling technology that potentially in combination with cell therapy could have explosive potential. But two, do you even need cell therapy? Is it the body's failure to recruit cells to the target? And so to explore those concepts is Rahul this morning. Thank you.
Thanks a lot, Jason. Hopefully you can all hear me. I appreciate the opportunity to share a little bit about Juventas with you and to discuss it with Jason in more detail. So just to give you a high-level introduction, we're based on technology that was exclusively licensed from the Cleveland Clinic around an IP portfolio developed by our founder, Mark Penn, cardiologist scientist, really grounded in the concept of identifying molecular factors that activate endogenous tissue repair. Our lead product is a gene therapy. It's a little different from the gene therapies you've heard about so far in that it's a non-viral DNA plasmid encoding a molecule called stromal cell-derived factor 1, and there's certain advantages to that non-viral strategy that I'll present to you. We've always focused on advanced cardiovascular disease with clinical stage programs in advanced ischemic heart failure as well as late-stage peripheral artery disease. We've advanced both those programs through exploratory phase 2 clinical studies, having demonstrated a very nice safety profile as well as good signs of efficacy, which are now propelling us into phase 2B studies. And we've done this around an excellent team that's really managed to execute on time and in budget, having gone all the way from concept in 2007 to being phase 2B ready in multiple large indications in less than $35 million raised. So the foundation of the company is around work that Mark started about 15 years ago, through which he identified a molecule called SDF1, which we now recognize as a natural repair signal that's released by just about every end organ system to help activate your natural repair pathways. So Mark was intrigued by the concept that if you put a stem cell in the blood of an animal, a day after a heart attack, it migrates to heart. If you do it 3 months later or a month later, it no longer does. So he set out with the hypothesis that the newly injured heart is releasing some factor that has the ability to recruit your own stem cells and therefore potentially repair that tissue. Fast forward 15 years, what we know is that key factor is SDF1, and through interaction with this receptor CXCR4, it can repair tissue through a number of key mechanisms. It does recruit these endogenous bone marrow-derived stem cells. They turn into new microvasculature. There's also a recruitment of tissue-specific stem cells, so cardiac myocytes in the heart, neurons in the brain, so there's a little bit of smartness to the system. Directly, we also have a trophic effect, preventing ongoing cell death, and are able to either prevent and remodel fibrosis in acute and chronic states. So it's really this preserved biology that we're being able to exploit primarily in the heart at this point. We have taken a non-viral gene strategy because Mark's early studies demonstrated that you needed about 3 weeks of transient expression. And we could get that by delivering a non-viral gene therapy, and that allowed us a safe mechanism in which to get the proper expression, and most importantly, not induce an immune response. And by doing so, what we've preserved is the ability to be able to reintroduce this therapy to patients in chronic progressive disease as their disease advances. And that's a very large part of our upcoming clinical studies. Commercially, it's highly scalable, highest cost of goods at our top dose is around $100. So it provides a very nice commercial characteristic.
Let's go back to that prior slide if we can. And I just want to talk a little bit about what is the cost of goods and time to production to make this JVS 100, which I characterize as a DNA-based plasmid.
So we joke, if you can make beer, you can make plasmid. It's a microbial cell bank. It is highly scalable. We're running 50-liter reactors right now. We've transferred to 2 GMP facilities. We're estimating that the cost of goods will be between $1 to $3 per mg, and our highest dose is at about 30 mgs.
And how do you deliver the plasmid so that the cells will take it and then it will cause the cells to express SDF1?
Yeah, thanks, Jason. So the key here is to create a beacon. So it's direct delivery to whatever tissue we're targeting. In the case of the heart, we use an endoventricular catheter-based delivery that directly places around the infarct region in the heart. In our vascular indications, it's a direct injection to the leg. We've even demonstrated dermally that if you topically apply it to the wound, you reduce scar formation. But by applying it to that local site, you create almost a GPS signal through which the stem cells can migrate to that.
And how do you control the strength and duration of the signal so that it will be active for a long enough period to actually recruit cells?
So, some of the beauty is in the simplicity of JVS 100, but a lot of work went into that. So we spent a lot of time optimizing this vector with promoters, enhancer sequences, and other things that allowed us what we identified as the optimal expression pattern to induce response. And what that is a peak expression around day 3 that lasts for about 4 days, and then it tails off with expression waning by about day 20. Importantly, what Mark had demonstrated early on is that constitutive expression, while it creates benefit, does not give you any greater benefit than this profile. We've also looked at expressing shorter or at lower levels, and we don't get this type of benefit. So there's been a lot of work into the vector backbone to maximize that profile.
And does this have to be delivered to the site that you're trying to recruit the cells? And the cells then, once they home on this vector, will they stay around long enough so that they will secrete their trophic factors in order to achieve I assume a revascularization or maybe toning down inflammation or adjusting scarring, or the idea is just to create the signal for the cells to come?
Yeah, so it really is, again, it's the beacon concept. So the key is to get it to the area of injury. And then as we say, they call in 2 stem cells, they call in 2 of their friends, and we start a process. Those stem cells release factors. In an acute setting, those factors tend to have direct trophic effects, prevent scar expansion, and reduce inflammation. What we're seeing in chronic settings is a significant impact on microvascular formation. And this helps resuscitate hibernating myocardium in the case of the heart, heal wounds in the case of late-stage peripheral artery disease, and creates a healthier tissue for which the cells can operate.
Let's keep going.
So the 2 areas that we have primarily focused are late-stage heart failure and late-stage Ph.D. We have some exciting data now from what's called the STOP-HF study, which is a phase 2, which we'll plan on releasing in detail probably late summer at one of the cardiology conferences. But what I can tell you is that we've clearly demonstrated a safety profile that in patients with advanced ischemic heart failure that we're seeing sustained benefits in cardiac function, EF, structure, so end-systolic volume, and NT-proBNP, which is a marker for cardiac congestion. What's comforting to me as I look at this, and I think what's got our KOLs very excited, is the confluence of the data and the consistency of the response across multiple factors. And based on this, what we're planning for is a phase 2B in which we'll focus on this advanced population, so confirm that they're the target group that's most responsive. We'll do it with the dose that was most effective, which was 30 milligrams. But we're also going to extend our observations by redosing these patients at a period 6 months after their initial. The thought process being that we can induce repair, but it's always been our philosophy that there's no magic bullet. So we're never going to cure heart failure with a single dose of anything in our view. And so the ability to come back and readdress these patients with the therapy allows us to either further extend or enhance the benefit that we're seeing. The other area that we're looking at is late-stage peripheral artery disease. And I think there's an important distinction of where we're going relative to where the field has been. So our first foray into this was with CLI, and I think most people know that as no option, poor revascularization candidate patients who who have really exhausted all the device interventions out there and are at risk of amputation. That's where we did our first study, and I can tell you, along with the field, what we've seen is that amputation rates have dropped from about 40% down to mid-single digits. And a large part of this is because of interventions in the device space that are now allowing these cardiologists to get all the way down to the heel and revascularized tissue, or the limb. But what we've learned through that process is while they can create arterial flow and help create limb salvage, they're not getting tissue, they're not getting blood to the tissue wounds, and they're basically lacking microvasculature. What they approached us with was this concept that we can create the macrovasculature, you can create the micro. Stop waiting until we're failed to come in with your therapy, but work alongside us. So we're proposing a phase 2B in which we'll look at patients with late-stage Ph.D. that are undergoing below-the-knee angioplasty or stenting, with the concept that they'll open up the vasculature. We will then inject JVS-100 in the area of the foot to help heal their wounds. And to give you an idea, at a leading clinical center that we're working with, one of the interventionists said they did about 200 procedures in the last year and a half. They had 100% limb salvage, but in that population, they had 17% wound closure at 3 months and about a third closed in a year. So the good news is these patients are keeping their limbs. The bad news is 9 months to a year later, they're still not walking. They suffer with incredible pain. So those are two studies we're looking to start in the second half of this year.
If I was going to ask any one question, and I always wonder about this, and I kind of alluded to it in the introduction, these tend to be patients that have multiple comorbidities. If successful and you create the homing signal, are there resident stem cells that can hear it? And doesn't this therapy lend itself to, in combination with, say, an allogeneic stem cell therapy?
So there's two answers to that. Where we're seen in a JVS-100 alone setting is that we do recruit endogenous tissue-specific stem cells. We can all debate what those stem cells actually do when they migrate to the signal, but they do end up there. Your point's well taken, and I can tell you that in our founder's lab, he's been cell agnostic and really driven towards mechanisms that activate and enhance endogenous repair through stem cell-based processes. In his studies, he's shown that combinations of cells plus JVS-100 significantly enhance the activity of that cell. So we have a belief, not only with JVS-100, but a couple of factors that we have in our pipeline, that we can make any cell perform better. However, our primary focus really is to develop JVS-100 as a standalone therapy, appreciating that the field may migrate to a point where they deliver both together.
Right. I mean, you would see what an incredible 1-2 punch that is if you create a signal that doesn't exist in congestive heart failure, and at the same time provide the patient with a bolus of robust, powerful mesenchymal cells that can then act on that signal.
So, and I shared a little bit of this data with you, I think, last time we talked, but we have another molecule called JVS-200. And it is a potent chemoattractant for mesenchymal stem cells. And what we've been able to show is if you depot the JVS-200 in the target tissue, you can turn allogeneic MSC therapy into an IV. You get significantly greater cell retention, engraftment, and function. So those are concepts, again, we're looking at activating endogenous repair. We have factors that enhance cellular response. It's really around distilling cell therapy down to the factors that are providing the greatest benefit.
So help me understand how, as a private company, you've been able to accomplish all of this. What have been your funding sources so far, and what are your plans for the future as we as investors, are interested in being part of this company?
So to date, it's a ridiculous amount of discipline. I mean, we've gone from concept through heart failure phase 2 and peripheral artery disease phase 2 studies in less than $35 million. I don't have a very fancy office. And we've done that with a key focus on following mechanism data and understanding the clinical populations. We're now at a point where we're thinking about larger phase 2B clinical studies. I think nicely we've proven safety and some preliminary efficacy. So while we've been venture funded to date and we're still looking at private sources of capital, we're evaluating the public markets. There's a lot of excitement around gene therapy. I think a lot of what's going on in the field around heart failure has people excited about that opportunity as well. And so we're putting ourselves in a position that if a public offering makes itself available and is the best path, we'll take that forward. We're also having very productive discussions with potential regional and global strategics that may or may not be mutually exclusive with any of those financing sources.
Thank you. And just close with me a little bit on what are the data catalysts that I should be focusing on over the next 12 months as you potentially march towards a decision, whether it's partnering or an IPO or other.
So we receive data every quarter for about the last year and a half. So this is the first time we don't have data coming in the next few months. But along those lines, we're now preparing for the next transition. So some of the major catalysts are going to be clearance from the FDA on phase 2B designs for both these indications. They're novel designs with therapies that I think in the gene therapy space introducing no concepts of repeat introduction of the drug and following that patient over time. I can tell you our conversations with the FDA, we were just with them on Monday, have been very positive. So we anticipate we'll have those studies in place. As far as data, assuming that we're able to start our studies on time, we anticipate we'll have Ph.D. data starting to flow in the second half of next year, and heart failure data after that. And again, by the end of 2017, early 2018, assuming the financing gets in place, we could be a company with 2 phase 3 programs in large indications with really no other therapeutic options.
So you should see what I see, which is paradigm shifting, because we could talk about athersis and mesoblast. Neostem is going to follow us, but the reality is that if you could use a small molecule, off-the-shelf, readily available therapy that can boost a homing signal and recruit the endogenous stem cells or the teleologic ability of the body to heal itself, then potentially you could obsolete the stem cells therapy space.
I'm a little biased, but I like the strategy.
Yeah,
Makes
okay.
sense.
But I also believe that synergistically in older patients with multiple comorbidities, what we're really talking about is just a very, very powerful set of technologies that puts in place the mechanisms for healing.
The only thing I'll caveat that with is our data from our STOP-HF study shows that there's no lesser response in diabetics or the elderly. But to your point, the combination may still be more beneficial. So that's something that the field will evolve over time.
Terrific. Thank you so much.
Yeah, thanks a lot, Jason. Good seeing you.